right, mitochondrial DNA. So mitochondrial DNA is the DNA that's not in the nucleus of the cell. It's kept outside the nucleus in the body of the cell in a small organelle called a mitochondria that is the power generating system for the cell. And it has its own DNA because there's a lot of activity going on there, and it just needs a set of instructions immediately available to it because of all the things that can go wrong with power generating. So now this is then passed on from mother to child without being mixed up with the DNA of the father. And that allows us to track these mutations. And the DNA mutates over time. Like anything else, it's wearing out and errors creep in. So it passes from mother to child without mixing with the father's mitochondrial DNA. Worldwide rates of change have been carefully measured. Oh, I got measured in there twice. I'm seeing double. And this means, great-grandmother, daughter, granddaughter, on and on, multiple generations. Indigenous populations around the world have all had their mitochondrial studied, and how long it takes to mutate, how quickly does mitochondrial DNA mutate. So if you compare the data, how similar or how different the mitochondria is from everybody on Earth and how fast it's changing, all you have to do is back up that time. And this gives a time frame of 6,000 years. So human beings have only been on the planet for 6,000 years based on the actual measurements of mitochondrial DNA mutation. This is an example of population dispersion patterns, migration patterns, done using mitochondrial DNA. These patterns match the, let me see if I can, oh, you can see my pointer. Match descriptions given in Genesis. Now, here's one thing about this. It's very easy, you can tell what direction people went if you have more than 2 data points. Right? If you have 3 data points, you're looking for the direction which has the most mutations. So the further away you move, the more likely you are to have mutations. So the most mutated people arrive down here. And it's not, anything racist or not. This is the facts. Now, if you're here, it looks like, well, it says it came out of Africa, but that's because before these measurements, they already believed in the out of Africa theory. If you go from the actual origin could actually be over here, right? You can't tell because you can't tell which direction. They're just different, right? This one is different from this one. You can only tell the direction if you have multiple locations. So this actually isn't necessarily the beginning. The beginning could be right here where these other people groups are breaking off. So one goes this way, one goes this way, one goes this way, one goes this way, one comes down this way. So, and that will match the Y chromosome data also, and even the nuclear genetic measurements that have been done. Okay, so there's more additional proof that not only the creation and flood, but also the Babel story is correct. So it matches the Bible's description in the Table of Nations in Genesis 11. Oh, here's an interesting side thing is that Eve's original mitochondrial DNA has been resequenced, right? We now know what Eve's mitochondrial DNA. This was actually done by the two geneticists that I've already showed you pictures of earlier, Dr. Robert Carter, a PhD geneticist, and Dr. John Sanford, PhD geneticist, inventor of the gene gun. Those men worked together and were able to resequence by, figuring out which were mutations and which were the original, and moving it back until they were able to determine the actual DNA sequence of Eve, which is a terrific thing for the future for medical problems. There's lots of mitochondrial diseases because of the mutations that are adding up. Here is Y chromosome haplotype. This is a little more clear to see that the originating source is the Middle East. All of these down here, these are all different haplotypes or gene sequences in the Y chromosome. And when bottlenecks occur, when small groups of population move away from larger groups, they take with them only a representative sample, only a few. And the further you move away, the more time you do that, the more chances you have of losing information, until again you get out to South America and everybody is very what's called homozygous. Right, the information that they share in the Y chromosome is all identical. If you go back, you see the original sources had more information until the most diverse population of Y chromosome males is in the Middle East, right in this region in the Middle East. When human beings moved into Africa, they took only part of the DNA information. When they went into Europe, only part of the DNA information, up into Siberia, down into Australasia, only part of the information. So, so this is how we trace ancestries, but you can see, once again, it points to the Middle East. Okay, evolutionary genetic clocks. What are evolutionary genetic clocks? DNA sequence comparisons between taxa to estimate rates of mutation. All right, DNA sequence comparisons between taxa, different groups of species, to estimate the rates of mutation. These are, by evolutionists, routinely calibrated to paleontology. They have to match them to what they believe happened in the fossil record according to evolution. And the reason they have to do that, why they have to constantly recalibrate them, is because they don't match. Different genes give widely different rates. So if you calibrate one set of genes, according to paleontology, you find out when you come to the next set of genes that they don't match at all. So then that has to be recalibrated. And then you test another set of genes in those different taxa, and it doesn't match. Different taxa exhibit different rates for homologous sequences, right? They should be similar, but they're different. Divergent states commonly disagree with paleontology despite being calibrated with it, as I just explained. All right, now we need a little bit of explanation about mutations and neutrality. Standard evolutionary doctrine states that most mutations are neutral due to the redundance of codons. Codons are just like the words in the sentence of the nuclear information. Presence of large amounts of junk DNA would be expected from evolutionary doctrine, right? Because it's just accidental mutations over millions of years. So most of it doesn't work, it's just junk. But recent discoveries falsify this view. corresponding codons actually help control expression. So the fact that there's redundancy is actually useful. It's not accidental. DNA shows pervasive biochemical functionality. The presence of junk DNA is a myth. The vast majority of the DNA is actually functional, which makes no sense in a evolutionary theory. This was a falsification of evolutionary theory. Then comes along the DNA barcode project. So scientists wanted to figure out if there was an easy way to, if you had some DNA, that you could identify what species the DNA came from. So they looked at, it was a 10-year study of DNA published by the journal Evolution, right? One of the leading evolutionary journals, obviously. January 2019. They examined 5 million organisms in 100,000 species. And here is what they found. By tracing the genetic information, here's what they found. All human beings came from 2 parents. Okay, that was disputed by evolutionists. Species show little variation. So within a species, genetically, they're very, very similar. Differences between species are, however, genetically vast. Now, that's not supposed to be true according to Darwinian evolution. You're supposed to have a gradual change from one species into the next. They're not supposed to be clumped together like islands in an ocean. In fact, 90% of all species also came from 2 parents. So they were able to identify that the genetic modification over time showed that they came from 2 parents and not from large populations of species. And this generation from 2 parents happened at the same time. Are you hearing me, people? Right. So elephants are descended from 2 original elephant pair, male and female elephants, and they happen to be have the same number, the same time as crocodiles that also came from 2 parents, as ostriches, right? All these different species which have different mutation rates, but when you trace them back, they all arrive at their 2 parents at the same time. This generation happened in the recent past. Now, if you use just the raw data and you look at the current mutation rates of these animals, and you put that in the formula and calculate back how long did it take until they got to their 2 parents, it happens to be 5,000 to 6,000 years. Right about the time, 5,000 would be about the time of Noah's flood. But they don't do that because they don't like the answer. So what they do is they, again, adjust it to paleontology. So they estimated that there must have been some massive event that killed everything living on the planet, virtually everything, down to just a few mating pairs about 300,000 years ago. Now, the problem is that geologically, we know of no such event that happened at that time frame. But this is what the data forces them to do. Because they are unwilling to accept the actual raw data that shows that all this happened. I thought I had the, oh, darn, I don't have the paper. Well, you can look it up, January 2019 Evolution Journal. Okay, Let's see, it is 10 o'clock. I guess we should go to question and answer. I've still got tons on genetic entropy. Let's see, let me just cruise through this. Genetic entropy. Geographic dispersion, languages, research on tracing languages across the globe, mitochondrial clocks, date to 6,000 years, how many mutations you're likely to have, mutations per generation, published in Nature Genetics, using our empirical rate to calibrate the mitochondrial molecular clock would result in an age of mitochondrial DNA. This is a study in humans of 6,500 years. And then the next sentence is, but no one thinks this is the case. So what did they do? They changed the data. They matched it up with what evolutionists told them must be true about human evolution.